Good morning, dear friends. Virkar Miss here. Today, I am going to teach you pharmaceutical microbiology subjects. This is the subject of your semester 3, that is second year. There are 4 subjects in semester 3: Physical Pharmaceutics 1, then Pharmaceutical Microbiology, then Pharmaceutical Organic Chemistry 2, and Pharmaceutical Engineering. Out of these 4 subjects, I am going to teach you pharmaceutical microbiology theory as well as pharmaceutical microbiology practical. The choice code for pharmaceutical microbiology theory is BP303T, and the choice code for pharmaceutical microbiology practical it is 307P. Before actually starting with our syllabus, I am going to tell you the scope and objective of pharmaceutical microbiology subject. It means why we study the pharmaceutical microbiology, or what is the necessity of studying this pharmaceutical microbiology subject, or what is the scope, or what are the objectives why we study this subject. That is why we study the pharmaceutical microbiology subject. What is the need of this subject in our pharmacy or in our pharmacy field? The scope of microbiology is to study the all categories of microorganisms. That is to study the all type of microorganisms or the all categories of microorganisms. For example, bacteria, viruses, fungi, parasites, yeast. These are some examples of microorganisms. To study the all categories of microorganisms, especially for the production of alcohol, then production of antibiotics, then production of vaccines, then production of vitamins, and the production of enzymes. काय म्हणले तू आय झालं मग हे केलेलं गेलं म्हणजे फोनच्यात जागाच नाही. आता हे काय करायचं काय केलेलं गेलं ह्यात सेव्ह झालं नाही, फोनमध्ये जागाच नाही. आता मेमरी कार्ड आणावं लागत. अर्धा तास केलेलं बाहेर गेलं. The scope of pharmaceutical microbiology is to study the all type of microorganisms or the all categories of microorganisms, especially for the production of alcohol, antibiotics, vaccines, vitamins, and enzymes. Then objective of this pharmaceutical microbiology is upon completion of the subject, the student shall be able to understand. The first objective, understand the methods of identification, then methods of cultivation, then methods of preservations of various microorganisms. Then to understand the importance and implementation of sterilization in pharmaceutical processing as well as pharmaceutical industry. To understand the importance and implementation of sterilization in pharmaceutical processing as well as pharmaceutical industry. In microbiology, the word sterile is most important. So what is meant by sterile? The sterile means it means it should be, or the object should be free from all type of microorganism. When we we say sterile if our object or if the product is free from all type of microorganism. Then and then only we can say that our product is sterile. And what is sterilization? Sterilization, it means it is the process of being the product sterile, doing the product sterile. It is the sterilization. Then third, to learn the sterility testing of pharmaceutical products. As I said earlier, importance of sterilization, or what is sterilization, or what is sterile. Sterile means free from all type of microorganism. This is the main objective of your microbiology, that to learn the sterility testing of pharmaceutical products. Then next objective, to carry out microbiological standardization of pharmaceuticals. To carry out microbiological standardization of pharmaceutical. And next, to understand the cell culture, then cell culture technology and its application in pharmaceutical industry. These are the few objectives of pharmaceutical microbiology subject. First objective, to understand the various methods of identification, cultivation, then methods of preservation of various microorganisms. The next objective, to understand the importance and implementation of sterilization in pharmaceutical processing as well as pharmaceutical industry. Then to learn the sterility testing of various pharmaceutical products or the various pharmaceutical formulation. Then next, to carry out the microbiological standardization of pharmaceuticals. Then next, to understand the cell culture technology and its to understand the cell culture technology and its application in pharmaceutical industry. These are the few objectives of studying the pharmaceutical microbiology. Then marking system for pharmaceutical microbiology, it is for theory, for 10 marks for your continuous mode assessment. Continuous mode, it means your attendance, assignment, test, seminars and group discussion. Continuous mode, it means the 10 marks for continuous mode. In continuous mode, the marking division is attendance, seminar, test, group discussion, quiz. Then 15 marks for your sessional examination. 15 marks for your sessional examination, sessional 1 and sessional 2. That is total 25 marks for your internal assessment. Internal assessment, that is for 25 marks. 10 marks for continuous mode and 15 marks for your sessional examination. Sessional examination, the mean of both sessional 1 and sessional 2, that is 25 marks. And for your university theory examination, it is of 75 marks. That is total of, it is of 100 marks. And for practical continuous mode, it is for 5 marks and sessional examination for 10 marks. In continuous mode 5 marks, the division is 2 marks for your attendance and 3 marks for your daily practical evaluation. And for sessional, it is the mean of sessional first and sessional 2. That is total 15 marks For internal assessment of microbiology practical, and for university practical it is of 35 marks. It is total 50. It means the continuous mode for 15 marks, in continuous mode 5 for 15 marks for internal assessment and 35 marks for your university practical examination. This is the marking system of your pharmaceutical microbiology subjects. Then we discuss the syllabus of our subject. There are 5 unit in your syllabus. Unit 1, 2, 3, 4 and 5. In unit 1, history, introduction, scope and objective of your microbiology subject. Then introduction of prokaryotics and eukaryotics. Then structure of bacteria, ultrastructure of bacteria, morphology of bacteria, then classification of bacteria, then nutritional requirement of bacteria, raw material for culture and physical parameters of growth, then growth curve of bacteria, then study of different types of different types of phase contrast microscopy, then dark field microscopy and electron microscopy, then different methods of your cultures, then cultivation of aerobes, then quantitative measures of bacterial growth, in that first total count and second viable count. Then unit second, identification of bacteria using staining technique. There are different staining techniques are used for identification of bacteria. First technique, simple staining technique. Second, Grams staining technique. Then acid fast staining technique and biochemical test. In that first imimic test. Then study of principles, procedures, merits, demerits and applications of physical, chemical, gases and mechanical method of sterilization. As I said earlier, what is sterilization and what are the importance of sterilization? The evaluation of efficiency of sterilization method. Then equipments employed in large scale sterilization, and different indicators or the types of indicators that are used for sterilization or sterility. Then in unit 3, study of morphology, then study of classification, then study of reproduction, then replication and cultivation of fungi as well as viruses. Then classification of mode of action of disinfectant, then factors affecting on disinfectant, then factors affecting on antiseptic and their methods of evaluation. Then that is the evaluation methods of disinfectant and antiseptics. Then काय माझ्याकडे थोडं then sterility testing of various products, that is sterility testing of solids, liquids, ophthalmic and other sterile products. The sterility testing as per Indian Pharmacopoeia, British Pharmacopoeia, and United States Pharmacopoeia. Then Unit 4, the designing of aseptic area, laminar flow equipment, study of different sources of contamination in aseptic area, then methods of prevention, clean area classification, then principles and methods of different microbiological assays, then methods of assessment of new antibiotics, then Unit 5, types of spoilage, the factors affecting on the microbiological spoilage of pharmaceutical products, factors affecting on the microbiological spoilage of or the microbial spoilage of pharmaceutical products, sources and the types of microbiological contaminant, assessment of microbial contamination and spoilage, then prevention of pharmaceutical product using antimicrobial agents, and the evaluation of microbial stability of formulation. This is the syllabus of your pharmaceutical microbiology theory. From syllabus here we can conclude that, or here we can say that in pharmaceutical microbiology the sterility is very important. Without sterilization is formulation is not possible. Sterilization and contaminations are very important. Generally, the pharmaceutical microbiology provides the knowledge and understanding with regard to the significance of the presence of bacteria, yeast, mold, viruses and toxins. It means it provides the knowledge and understanding with the different types of microorganisms in pharmaceutical raw material, then intermediate products and pharmaceutical production. The word microbiology is derived from Greek word that is micros, it means small, and bios means life. Microbiology is derived from Greek word. In Greek, micros it is small and bios it is the life. It means it is the branch of science that deals with the study of the biology of microorganisms and their effects on other living microorganisms. For example, bacteria, protozoa, algae, fungus and viruses. The microbiology, it may be applied microbiology or the pure microbiology. First, pure microbiology, it includes bacteriology, mycology, protozoology, phycology, parasitology, virology, nematology, cellular microbiology, evolutionary microbiology, and molecular microbiology. All are comes under pure microbiology. I am once again repeating. The pure microbiology, it includes bacteriology, mycology, protozoology, phycology, parasitology, virology, nematology, cellular microbiology, evolutionary microbiology, and molecular microbiology. Then second, applied microbiology. It includes medical microbiology, pharmaceutical microbiology, industrial microbiology, food microbiology, soil microbiology, water microbiology, air microbiology, and microbial technology. I am repeating, applied microbiology. It includes medical microbiology, pharmaceutical microbiology, industrial microbiology, food microbiology, soil microbiology, water microbiology, air microbiology, and microbial biotechnology. When the microbiological concepts and the microbiological processes and the microbiological techniques are applied to the pharmaceutical operation, then the subject is then called as pharmaceutical microbiology. When the microbiological concepts, the microbiological processes, and the microbiological techniques are applied to pharmaceutical operation, then the subject is then called as pharmaceutical microbiology. The main objective of pharmaceutical microbiology is to ensure the safety and efficacy of pharmaceutical products. The main objective of pharmaceutical microbiology is to ensure the safety and the efficacy of your pharmaceutical products or the pharmaceutical formulation. It embraces to validation of disinfectant, of the evaluation of the efficacy of the disinfectants in suspension on surfaces and through free field trials. It offers the protocols and techniques associated with the operations and assurance of clean room, aseptic room, and controlled environment for preventing any possible microbial contamination, and introduces risk assessment and practical contamination control the strategies. The Louis Pasteur is a scientist. It is regarded as a father of microbiology, and whereas the Robert Koch is the father of medical microbiology. There are two different scientists. First, Louis Pasteur is regarded as a father of microbiology, and second, the Robert Koch is regarded as the father of medical microbiology. There are two different terminologies: father of microbiology and father of medical microbiology. The father of microbiology is Louis Pasteur, and the father of medical microbiology it is Robert Koch. First, in history, in 1673 to 1723, the Antony van Leeuwenhoek, that is Dutch, he described the live microorganism that he observed in teeth, scraping, and rainwater. And he is the first person to actually see the living microorganism. Then first, Louis Pasteur, it is the French organic chemist. He is considered, as I said earlier, he is considered as a father of modern microbiology. He said that the fermentation process, it means the beers and wines are not produced without microbes. The fermentation process, that means the beers and wines, the production of beers and wines is not possible without presence of microbes. Then the microbes causes the spoilage. Then Louis Pasteur has also demonstrated the gun cotton experiment. Then he also performed the swan neck flask experiment. Then different places and the different concentration of microbes. Then Robert Koch is the German physician. As I said earlier, he is considered as a father of medical microbiology. First, he demonstrated the role of bacteria in causing diseases. He first isolated the Bacillus anthracis, then discovered the Mycobacterium tuberculosis in 1882 and Vibrio cholera in 1883. He awarded the Nobel Prize in 1905. The Robert Koch developed the relationship between the microorganisms and diseases. Most important, that is, Robert Koch developed the relationship between the microorganisms and diseases. There are few postulates of Robert Koch. The first postulate is the microorganism must be present in every case of disease. The second postulate, the microorganism must be isolated from the diseased animal and grown in pure culture. Then third postulate, the disease must be reproduced when the pure culture of microorganism is inoculated into susceptible host. And last postulate, the same microorganism must be desolated from the experiment from the experimentally infected host. I am once again repeating the postulates of Robert Koch. The first postulate, the microorganism must be present in every case of disease. This is the first postulate. Second, the microorganism must be isolated from the diseased animal and grown in pure culture. This is the second postulate. Then third postulate, the disease must be reproduced when the pure culture of microorganism is inoculated into susceptible host. Then last, the same microorganism must be desolated from the experimentally infected host. These are the postulates of Robert Koch. Then there are few branches of microbiology. First branch, protozoology, bacteriology, mycology, parasitology, immunology, virology, phycology and nematology. I am repeating the branches of microbiology: protozoology, bacteriology, mycology, parasitology, immunology, virology, phycology and nematology. What is microbiology? The study of living organisms of microscopic size. Microscopic size, it means those microorganisms are visible only under microscope. It includes bacteria, fungi, algae, protozoa and viruses. In microbiology, it also deals with structure, morphology, reproduction, physiology, metabolism and classification of microorganisms. Then microorganisms have both beneficial and detrimental effect on environment and human welfare. For example, yogurt and cheese making, lactobacillus and streptococcus bacteria. Then wine making, it is Saccharomyces cerevisiae, it is yeast. Then penicillin production, it is Penicillin notatum, it is the example of bacteria. Then disease causing bacteria, Vibrio cholerae, it is the example of bacteria. Then spoil food, it is the example of fungus. सचिन थांब घे, मी बंद केलं रेकॉर्डिंग. इकडे मला मेमरी कार्ड पाहिजे बा, यात फोनमध्ये काही स्पेस नाही. मी अर्धा तास रेकॉर्डिंग केलेलं पाहिजे. अर्धा तास रेकॉर्डिंग केलेलं अनसफिशियंट फोन मेमरी मध्ये. केवढं मोठं आणलं त्या फोनमध्ये बसेल तेवढं. हे 4 GB असेल, 8-10 GB बसेल की. ह्यात बसेल का? त्याला विचार Y51 मध्ये कितीचं बसेल. आजच्या झाले मला मेमरी कार्ड दे फक्त, फोन राहते बोल. अर्धा तास झालं रेकॉर्डिंग वाया गेलं माझं. Then deterioration of material like iron and wood by bacteria and fungi. Example, yogurt and cheese making, lactobacillus and streptococcus is the example of bacteria. Then wine making, Saccharomyces cerevisiae, it is the example of yeast. Then penicillin production, Penicillin notatum is the example of bacteria. Then disease causing bacteria, Vibrio cholerae, it is the bacteria. Then spoiled food, it is the fungus. Then deterioration of material like iron and wood by bacteria and fungi. Then microorganisms in the living worlds, plant, animal and protista. Then protist, bacteria, algae, fungi and protozoa. Then groups of microorganisms. First, bacteria, it is unicellular prokaryotic organism or association of similar cell, and the cell multiplication of bacteria, it occurs by binary fission. What is bacteria? It is unicellular prokaryotic microorganism or the association of similar cells, and the cell multiplication of bacteria is occurs by binary fission. I will stop here, but one question on this lecture. Write about scope, objective, and history of microbiology. This is the question which is based on this lecture. Those student submit the answer of this question, then and then only the attendance of this lecture is considered. If those students are not submitting the answer of this question, then attendance of this lecture is not considered. It means the student are not submitting, he is considered as absent for this lecture. The answer is submitted via WhatsApp or email, or the answer is submitted via WhatsApp and email. I am once again repeating, the answer is submitted by the student via WhatsApp and email.